The Sydney Brenner Institute for Molecular Bioscience is a Wits University Genomics Research Institute and a centre for learning for young African investigators. Our scientific team guides large African studies that all involve genomic data from African populations. This provides the basis from which to include African individuals in international databases. Our team of 5 senior investigators lead studies on African population genetics and the bioinformatic analysis of large genomic datasets.
So the aim of our research is to identify the genes involved in common African cancers, particularly breast cancer and esophageal cancer. We scan the entire genome of thousands of African patients to find the genes which increase their cancer risk. And we use DNA sequencing to look at tumor tissue from the patients to identify the rogue genes which are driving the development of their cancer. We are building a team of young African scientists who will provide scientific support for a precision medicine approach to cancer diagnosis and treatment in South Africa.
The focus of my research is to generate a fine-scale map of the genetic landscape of the continent and to estimate how past events such as migration and intermingling of people, as well as factors such as climatic fluctuations and pathogen exposures, have uniquely shaped the African genomes. So my work serves as a scaffold that enables other members of our team to better design their studies and also suggest them the optimal analytic framework to conduct their work.
Bioinformatics is the application of computational techniques to solve problems in biology, and it's the heart of modern genomics. At the SBIMB, we apply state-of-the-art computational techniques to solve problems in population genetics, as well as finding genomic variations that might be implicated in disease. We're also active in building novel bioinformatics techniques and building computational infrastructure. We hope this will form the basis of precision medicine, tailoring drugs and treatments to different populations and individuals.
Together as a team, we address genomic risk factors for disease and the role of genetic variation in treatment response, thus enabling us to contribute to Africa's vision for genomics-driven precision medicine. Through targeted gene, whole exome, and whole genome sequencing, we continue to show that African populations have greater genomic diversity than other world populations, such as those of European and Asian ancestry.
Our goal is not only to do great science, but it's to grow the next generation of genomic leaders on the African continent.
I feel very privileged to be a part of a team that is contributing to the genetic mapping of our continent because it feels like I am working on my future.